Good evening, everyone. A very warm welcome to every one of us. You joining us today for the session 62 of the Beyond Research, where curiosity meets conversation. We are delighted to have you with us for another inspiring discussion that brings cutting edge science closer to the community. Today, the session is titled Seeing Life in a Frozen Snap, Cryo-Electron Microscopy for Biological Discovery. Cryo-EM has transformed our ability to visualize the molecular world with uh, with extraordinary detail, and we are thrilled to explore this powerful technology with an expert in the field. Welcome, Dr. Pankhuri Narula. So, our speaker for the day is Dr. Pankhuri from the Kusuma School of Biological Sciences, IIT Delhi. Dr. Narula completed her PhD at IIT Delhi in the area of peptide therapeutics during her postdoctoral tenure. She stepped into the fascinating world of the cryo-electron microscopy, working extensively on non-enveloped viruses and studying how they disassemble, an essential step in establishing infection. Currently, her research focuses on capturing and understanding the transient disassembly transition states of viruses, a direction that holds great promise for the development of future antiviral drug molecules. We are honored to have you with us, Dr. Pankhuri. With that, I would now like to invite you to begin the session, and over to you, ma'am.
Hi, good evening to all. Thank you so much for such a kind introduction. And I'm very excited to just talk about this new technique in the field of biology. And I hope some of you must have read about it, but I will do justice to it by giving an introduction and how it is doing rounds of new discoveries and breakthroughs in our field. So if I think you have given me the permission to share.
Yes.
Okay. Okay, so is it visible?
Uh, yes, it is visible.
Okay, so should I start?
Yeah, you can start.
Okay, so majorly what I'm going to talk about in this meeting is uh, a very breakthrough technique and how it is doing a round of changes in the field of biology. So I have given the title to my talk as Seeing Life in a Frozen Snap, and I think I'll be able to explain this whole meaning in my upcoming slides. And how do we see life in a frozen snap is through cryo-electron microscopy, and specifically using it in the area of biological discovery. So Vinita has already given an introduction about me, so I'll just brief you again. So I work in the area of virology. So I got a chance to explore this technique when I stepped into my postdoctoral part, where I was given a task to work on the non-enveloped viruses and try to capture virus at a stage in its life cycle where it is transitioning, where it is uncoating and releasing its genome. So we are trying to capture it and we are trying to structurally analyze it. So that I'll talk at the end of the my talk, just again to tell you about my research focus. But majorly in this present slides, I'll be talking about the cryo-electron microscopy, its history, how, what, when, and everything about that revolves around it. So I will just start with the basic thing like electron microscopy. Why electron microscopy? So if we segregate into the two words, so electrons here stands as a source, which are used as the beam of electrons when they fall on your test sample or the specimen. They interact with it, they are absorbed and diffracted in different patterns. Those patterns are generated, they are analyzed digitally, and then they form an image which gives us an information about the test sample or the specimen of interest. Microscopy, as you all know, is the magnified image of the objects which are not visible to the naked eye. And if you'll see here the scale, just a second, I'll just switch on my pointer. Okay, so if you'll see here the scale, our naked eye can typically see the fungus. You can see it everywhere around you. the mycelia. But if you want to see the objects or the organisms which are smaller than that, you will need a light microscope. Light microscope works till 1 micron, but which through which you will be able to see the bacterias. But if you want to see the more atomic details of the viruses and the cellular organelles and other structures, you really need to have something called as electron microscope. So electron microscopes, it achieves a very high resolution because electrons as such has a very low wavelength. So it helps us in visualization of the things that are not able to see in the light microscope. And the application generally revolves around visualization of the viruses, the cellular organelles, the DNAs, the RNAs, etc. So overall, the electron microscope is scanning electron microscope and the transmission electron microscope. Unfortunately, both of these are available with us in the IIT. So as the name suggests, the scanning stands, it generally scans the surface of the sample. And the transmission electron microscope, again, as the name suggests, it transmits into your sample and gives detailed information about what is inside the sample. So SEM typically requires a coating of the conductive material, whereas for the TEM, you really need to have a very thin slices where it is very easy for the beam of electrons to penetrate and diffract and give us a pattern. SEM kind of has a lower resolution, but TEM has a very high resolution. And again, SEM is used for studying the surfaces, and the TEM is again for the getting a detailed information about what lies inside the cell. So here I'll be talking about the transmission electron microscope. So if you really want to imagine the life inside the human cell, where everything exists in an atomic level. So imagine you being, you are just shrinked to such a level that you jump inside the human cell, and then you'll see how the things interact, and there is a traffic, there is a flow of proteins and everything is running. There's a DNA production, there's the RNA happening. So the life is very complicated, yet disciplined, yet beautiful when you go inside the human cell. So everyone wants to unravel at different spheres what is happening, how this is so disciplined that everything is falling into place and so targeted. So there, these techniques help us to, kind of segregate to to have a detailed view of our interest, our level of interest where we want to see. So I will just give you, I have a very beautiful video. I'll just give you, uh, I'm not sure why it is not working. It was working till now. Let me see. Okay, it's working.
So let's say that this.
Okay, so this video, I'll just give you a brief. This revolves around how respiratory diseases, how the viruses, they spread. So once this is over, I tell you the relevance of showing this video to you. Just a second. Oh, it worked right now. I just hope. Anybody has any idea? What should I do? Okay, it's working. Right.
So let's say that this guy has the flu, could be any flu, and here's a droplet from his sneeze containing, if we move in and take a really close look, you see each one of those little purple things is a virus. And there are a lot of viruses floating through the air, some of which go inevitably up this unfortunate man's nose.
How did that guy feel when you ripped off half of his face?
It was interesting because we did it while he was sleeping.
Okay, I am talking with medical illustrator David Belinsky, who designed this video for Xyrus, a research company. So here comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things. So you call these keys, right?
Those are the keys.
Yeah, this is a key, this is a key, this is a key.
Okay, if the keys on this virus happen to fit the locks, which are those little purpley stick-uppy things on the surface of the cell, if there is a match, the cell, watch this, welcomes the virus in. And what's this?
This is the welcoming committee. They all interlock with each other and they pull this membrane down into the cell.
And down it goes deeper and deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses in those little noodly things. So this unsuspecting cell has been tricked into guiding these virus recipes right into its own command center, the nucleus. So in they go.
And they are immediately recognized by this big pink molecule, which is a mini factory.
Yeah, what is it doing?
It threads the nuclear material, the instruction code of the virus, through one hole, and out another hole comes a brand new instruction set.
So it's a copying machine making copy after copy after copy of virus recipes, which then go out of the nucleus to little chefs, those blue peanutty things. They cook up proteins that go back into the nucleus where they are reassembled into baby viruses, and then out they go. They get covered up and head to the surface where they get new keys, and then boom, here they come. This is an eruption of virus after virus after virus after only one virus entered the cell. But how many came out? Well, millions. So if one virus can produce 1 million babies and do it again and again and again, how come this guy doesn't just drop dead, I don't know, in like 10 minutes?
Well, because you have about 100 trillion cells.
I see. So 1 million viruses is just a drop in the bucket when you have 100 trillion cells. And anyway, remember, you do have your own immune system, which when it sees a virus, usually kills it. So while the virus does multiply fast, with any luck, your immune system will work just a little faster. So yes, all viruses want to spread. That's what they do. But most of the time, we do keep them in check. Most of the time.
Okay, I hope you find it interesting. So what I wanted to tell you from this video is, you see how there are keys on the viruses, right? So in case of the COVID, those keys are the spike proteins. So actually those are the keys which fit into the specific locks. That is why COVID was a respiratory virus and it didn't go to the kidneys and attack there. So there were the keys, and the key structure was solved by the cryo-electron microscopy. And there they found if they hold those keys in a specific conformation, they'll not be able to bind to the cells, and then there'll be no virus infection, and then there'll be no virus babies, and then it'll not spread. So that is how the COVID vaccine was formulated and designed using the help of the cryo-electron microscopy. So it is not just the viruses where this technique is used. You can see it is used in terms of the DNA and the uh, protein complex, which is the nucleosome. I think if you're from biology, you know this nucleosome complex, how they interact. Then, of course, the viruses. And then there are channel proteins, which are very important in case of the human cells, as they maintain this anions and the cations balance, which is of utmost importance when in diseases and everything. So I'll move on to the cryo-electron microscopy now, and I'll tell you the history. So it all started in 1970s, but biology was nowhere in the scene. So it started for the material sciences sample, that they are very strong and then they are very resistant to the environment and other factors. But then the scientists thought of, why not can't we use it for the biology and study life? And when they decided to do so, there were a lot of challenges that they faced. I'll discuss about the challenges. So, but it started with 1970s. The biology of cryo-EM started a little in 1980s, when Jacques he thought of some ways how we can, handle the biological samples so that we can study it with the use of the cryo-electron microscopy. And then in 1990s, the Mr. Frank came into the picture, and then he started some computing softwares where he wanted to really know how, if we get this set of images, how we are going to analyze them. And then in 1995 to 2000s, these challenges were addressed, and In 2012 to 2015, there came a wave of resolution revolution where there was tremendous updates in terms of softwares, in terms of technical specificity, technical specifications, in terms of cameras, in terms of sample purification methods, and it just revolutionized the way how we can use the cryo-electron microscopy for the biology. So if you see here, I was talking about the challenges. So the first was, in case of the biology, the sample specimen hydration was the first thing that came into their mind. And it used to get dried up, and it doesn't then be in its native form, which everyone wanted to see how the proteins and everything, they are in their native form. Like the native form is how they're behaving inside the cell. And then there was the ice formation. The water crystals were hampering the electron diffraction pattern. There was the radiation damage because of the electron beam. And since biological samples, they are just all carbon, hydrogen, oxygen, so they did not create a very good contrast to noise ratio. Sample preparation was again a problem. And then there was cost and technical things. So a lot of these things that were, addressed during 1980s to 2000s. And then this came the resolution revolution where vitrification was used. So they thought of to treat the sample uh, in such a manner that there was no water crystal. There was an ice formation, but the water doesn't crystallize it. So it is called the amorphous ice formation. So in this, you treat the sample in a very low temperature, around minus 200 degrees Celsius, and it has to be in milliseconds. And the technique is known as vitrification, that is the rapid freezing of the samples in the vitreous ice to prevent the crystal formation. Then everything needs to be done in the cryogenic temperature, which enables the longer exposures and the better image qualities. There were improved electron optics. Then there come the DEDs, which is the direct electron detectors. They're high sensitive cameras with faster frame rates and less noise. And then there was single particle analysis with with the promotion and the release of many softwares such as Relion, CryoSpark, and which enabled the processing of the images in such a manner that they were quite, they replicate the images that were formed after the this microscope analysis. Okay, so do you see this changes now? So this is, you can go through the years. So this is kind of a blob kind of a thing that you could see in the early cryo-EM years, and then it was refined, where you can see some changes on the surface, and more refined where you can see some chains and everything, and then it goes more refined where you can actually see the beta sheets and the alpha folds and everything. So from 17 angstrom resolution, it was brought to less than 3 angstrom with a period of time, and with the advancements that revolved around too many spheres of like computing the sample prep, the electron beam, and everything that revolved around that they could do. So after all this, so these three guys, Dubochet, Frank, and Henderson, they were awarded the 2017 Nobel Prize for the cryo-electron microscopy. So the Henderson pioneered the use of electron microscopy to visualize the proteins. So bacteriorhodopsin was the first biological molecule that was structure came out using the cryo-EM. So the Frank developed the image analysis method that computers assemble in a high resolution 3D image from 2D images that are collected. Tells you how the vitrification works. So this is a chamber. This is the liquid nitrogen minus 180. This is actually how we prepare the samples. This is, we're filling the ethene pod. Everything needs to be done at the cryogenic temperatures. These are the prerequisites that we do before our sample. This is the grid box where we store our grids. And this is the vitro boat where this everything is happening. This is more or less automated. This goes into the chamber which maintains the temperature and the humidity. This is the chamber. Here is the grid. He's putting the sample. This is the tweezer. Here's the grid sample, and these are the blot papers which will remove the excess sample. So we let the sample absorb onto the grid, then we blot the extra sample, and then do you see the amount of time it took to get plunged into the liquid ethane? And it's done. And this is how we store the grid. Many times it just fell down into the ethane pot. So there are some troubleshoots that we do. That's the everyday affair. So then they are ready to be screened by the microscope and the further processes. The whole process needs to be done at the liquid nitrogen level. If anything comes in contact with the air, the sample is gone. So I'll just quickly move you through this pipeline where every step has its own challenges. So if I'll talk about the sample purification, so if you're working with the proteins, you need to make sure it doesn't contain anything other than your sample, because that will lead to the heterogeneity and then it will impair your sample purity. So you really need to be very specific and very sure about what you are taking to put on the grid. If there is anything which you think is doubtful at the sample purification step, you need to go back to the bench again and again and again and work on the biochemistry. And once you're very sure your sample is of high concentration, is very pure, then you move on to the bench in the cryo lab, where you take a grid. Grids are again of various types, and it depends on your sample type which you want to analyze, which grid you want to take. There are high conductive grids, and then there are graphene grids, and then there are different sizes of whole grids. So you take a grid, you glow discharge. So generally, glow discharge is a technique which makes your grid ready for the application of your sample. Again, the glow discharging depends on your sample type. Sometimes you glow discharge or coat the grid with a certain chemical, which, like for example DNA, it is negatively charged. Of course, if your grid surface is positively charged, it will go and bind to it. But if your grid surface is negatively charged, there will be a repulsion. So you need to be very sure about your sample type and the type of the grid that you are using. So once you are sure about your grid type, you glow discharge it, and then you put it your sample on the grid. This was shown in the video. A tiny droplet as small as what 3 microliters will give you enough information on what is happening inside the cell. So that is a very much advantage of cryo, that you do not need larger volume samples. So 3 microliters is enough. It is just like a tiny drop. So after you put the sample, you just let it be there for a minute or so that it gets absorbed onto the surface, and then you blot the extra ones. So once your blotting is done, this is the vitrification, that is the plunge freezing. So you freeze your grid in as short as nanosecond or millisecond into the liquid ethane, so that your sample remains in its native conformation and there is no time for the ice to form the crystals. So this is how your sample will look onto the grid. Like this red are your protein samples, and then you take it for the imaging. There's the electron beam that gives kind of a patterns, and that pattern is digitally analyzed, which gives you a map that something is there on your grid which looks like this shape. And then you take it to the computing models, Reliant and Cryospark, and then they give you the detailed information about the atomic details, about the side chains and the amino acids and the beta loops and the alpha helices, everything. Okay, so if we'll talk about the crime applications, so it is varied in the biology area. So to start with structural biology, so it helps us to analyze the 3D structures of the proteins and the complexes, which in turn helps us to understand the mechanisms and the interactions and the conformations of the proteins. And if I talk about the virology, I've already given you the example of how cryo-EM has benefited the COVID research. And not only the complete viruses, but in fact the viral proteins, like the spike protein. They took out the spike protein and then they elucidated the structure. And then there is antigen antibody complex, where immunology comes into the scene. So all this aids in the vaccine design, the antiviral drug development, and it gives us an understanding on the viral life cycle. So this is the part where my study is focused. If we talk about the drug discovery, so it helps with in determining the targets where the specific microbes go and attach themselves, and then they can screen for the certain small drugs and analyze the structure, how they are reacting in a way. Neurodegenerative disease, again, cells is there's another thing which we call as cryo-ET, that is the tomography, which is you take out the cells and then you do the thin sectioning and then you analyze what is happening inside the cell. So it will kind of give you an information on the native environment of how the proteins are behaving or how the viruses are behaving, the absorption, the formation of the bulge, and then the uncoating and everything. So these are the different areas where cryo-EM is currently being used for in terms of biology. And in detail, if I want to tell you how it helped with the COVID, so There was the spike protein identification, which is the prime factor for the attachment of the virus to the cells. So it is a surface protein that mediates entry into the human cells, and it is a target antigen for the vaccine development. So this structure was determined using the cryo-EM. High resolution structure was of 3.5 angstrom was provided, and then they were able to see how the spike is oriented, what is its shape, and what are the domains that are getting attached to the receptor. So they found the RBD receptor, which is there for the spike interaction, and they were able to stabilize the spike in the pre-fusion conformation. Like before the fusion, there is a conformation of the spike protein. So when it attaches with the RBD domain, it changes its conformation a little so that there is a key and a lock mechanism. So when you try to hold spike protein in its pre-fusion state, it will not be able to bind to the human cell, and then there'll be no infection, of course. So a lot of vaccines by Pfizer and Moderna, they were based on the structure of the spike protein, and that is how we were able to get our COVID vaccine. And this is one example, and a very, I think, very relatable example where we can see the cryo-EM in the vaccine development. Another one, if I'll talk about the neurodegenerative diseases, if you know, in Alzheimer's, there is the tau fibril, and there's this conformational change, and the which is there in the Alzheimer's. So it has cryo-EM has helped to solve the structure of these fibrils and the other also pathogenic fibrils like synclunin and amyloids. And in addition to the cryo-EM, the cryo-ET has also enabled us to visualize the beta amyloid and the this tau fibrillar assemblies, and these have given us the understanding of the disease mechanisms by telling us how they are architectured, and how they how their structural and is linked to the disease progression. So yes, and then if I'll go forward, they have been involved into the replication stage, where they are the G-cord reflexes, which are stalled replisome, and they are kind of involved with the cancer strategies. And then there are this membrane repair proteins, and a lot of them. These are some of the examples I've thought of discussing where cryo-EM is involved. And this is just the overview of now what I have told you till now. This is the blotting.
Frozen in liquid ethane so rapidly
Ethane.
that ice crystals cannot form, and the protein molecules are held in their natural shape in a very thin sheet of ice.
So these are the protein molecules, and these are in different orientations.
Detected by a camera to produce an image.
Look,
The molecules in the sample are in different orientations. Application of a computer program sorts the images and groups all those protein molecules that are in the same orientation. Thousands of images are required for each sample. Further software is used to piece the images together to produce a high-resolution 3D structure of the protein. This is gamma secretase, a protein important in Alzheimer's disease. The structure of gamma secretase was determined by scientists at the LMB using cryo-electron microscopy. Being able to visualize a protein at this level of detail allows us to better understand how the protein functions. It opens up the possibility to design drugs that precisely fit proteins and offers an exciting opportunity to treat diseases.
okay, so I'll tell you what was happening in the video. So you put a sample, say in case you have a proteins. Of course, proteins can be organized into different orientations. Whenever you put a sample, those proteins will be fixed in those orientations. For example, out of 10, 2 are fixed in same, 2 are in same, 2 are in same. So what the cryo-electron microscope will do is it will give you different classes, like how many orientations it has seen and how many number of particles are there in that specific orientations, and then you club them together, you average them out, and then it gives you one 3D full map of what it has absorbed of all the particles that it has seen. And then the map is analyzed in the form of a projection, and then the projection is taken for the density analysis, and then from there you get your 3D model and you analyze the structure of your test sample. So we have with us Thermo Fisher's Titan Cryos G3, which is a very updated model of the electron microscope. It is placed in 99B building, and it was acquired with the help of the Sathi DST, and it is currently up and running, and it is giving beautiful structures of viruses, proteins, and other uh, cellular organelles. So if any of you want to come and visit and see, you can definitely contact me and Professor Manideepa Banerjee, and you can have a view. It is a beautiful microscope, and I think everyone should see it. And IIT Delhi is very fortunate to have it with them. And so that's it. I think this is it. And any questions, anything, I'll be very happy to take up. Nothing? Thank you, Azhar. Any curious or any question which is struggling in your mind is welcomed.
Yeah, this is a chance to ask your questions, dear participant. You don't hesitate. You can either put it in the chat, and you can also interact with ma'am. You can turn on your mic.
Sure. So I've got a question. I'm first year biotechnology student. Which skill I need to learn now? There are a lot of skills available. It depends on your uh, interest, the level of interest. There are microscopies. There are very, I mean, the AI biology thing that is very up and going in this time. So I mean, it is very much dependent on where your interest lies, rather than for me to tell. You just need to explore, which is very important. Any of the presentation question is also welcomed in terms of biology. See, if you want to read more about the cryo-EMs and the protein-based drug analysis, so the Richard Henderson belongs to the MRC, the Cambridge lab. So you can always, there are beautiful videos available online where he explains this technique in his own words. You can always go and learn from him. And there are a lot of lecture series on the YouTube present in relation to the cryo-EM, which explain the fundamentals of this technique. So you can always go and first learn the basis, and then you can learn how to employ it in terms of different aspects. So there is now a lot of videos available in YouTube and the MRC, the Cambridge lab specifically videos. So go and just watch them out, and then you'll know how beautiful this technique is, because it's an amalgamation of maths and science and biology all. And of course, this PubMed and everything for the latest research articles, you have to do it. Just type in the keywords and see what is latest in the field. Because what I could say is this is very recent. I mean, if you'll go back, this is what 2017 is very recent, and from there on it is 2020s when the computer softwares were developed and advanced to have the more updated version of the structures that we can get. Okay, we have Satya Deepika. I'm doing masters in biotechnology. Can you please give? Okay, so if you want for the industrial, so for the academy and the industrial, so there are two different streamlines, right? And if I'll talk about the academia, you really need to go into the research and everything. But if I'll talk about the industry, there you really need to work upon a skill set, right? Because industry needs, if they want somebody for the protein purification, you need to work upon the techniques which are involved in the protein purification. They don't want you to do any kind of a cell-based assays or anything. So if from now your mind is you have made up your mind to go for the industrial thing, you please start working in acquiring the skill sets, and you first zero down upon the industries you want to work up in, like the vaccine industry or the biologicals industry or the protein industry, or the dry labs, the computational industries. So you jolt down the zero down the industries where you want to work on, and then you can start working on acquiring the skill sets that aligns with those industrial needs. In academia, you are very much open, you are very much free, you have all the freedom to explore and research, and of course it's time taking, but if you're sure of going to the industry, you have to acquire a skill set and then you move on. I think I'm able to answer. Just a second. See, dark field microscope and bright field microscope, they are very, I mean, they just use as light as the source and then they produce the image as such. So I don't think there is a lot to explain in those dark field and bright field microscopes. So they are very simple, and it uses the light as a source, and then they illuminate the specimen, and with the formation of the contrast and everything, the image is formed. So I think you can. There are a lot of study material available that you can go and check in case of the bright field microscope. Fluorescence microscope is totally different. So fluorescence microscope requires a material that fluoresces. So fluorescences is what? Again, so when the fluorescence material is like, it emits the fluorescence when it releases its energy, right? So there are some fluorescence tags, they emit in that wavelength where you can see the way where you can observe the fluorescence. So they have the filters like for different wavelengths. If you will talk about the fluorescence microscope, we have the epi microscope, and if your protein fluoresces in the green zone, so it is like you see the green fluorescent protein, so it will lie in a certain kind of a wavelength. For the red it is 550 and beyond, for the yellow it is 488 and 500. So it depends on what wavelength your test material or the specimen is giving the fluorescence at, and then you can record it and see your sample. And of course fluorescence microscope doesn't use electrons as a source, no. It uses the light as a source. But you know what, there is a technique that is upcoming which people are working, the scientists are working in developing, which combines the fluorescence microscope with the cryo microscope, where they can tag their protein, and then they can see how it is reacting inside the cell. So you can locate your protein, how it is going, where it is going, and everything. It's the life tracking system. So fluorescence generally allows you to track the location of your test sample. So when it gets combined with the cryo-electron microscope, it can give you a beautiful high resolution picture of where your protein and how it is interacting with its substrate. I'm sorry, I don't have the expertise in stereo microscope and the freeze fracture. Okay, just a second. How can cryo-EM be employed for novel enzyme binding site identification? Okay. And efficient breakdown or cascades initiation. How can it help in designing a drug-like molecule to fit in the binding site? Okay. See, you know, a protein which goes and binds to a specific enzyme site, right? So, and then you know a disease in which that protein is distorted or it is get conformationally altered, right? So what you can do is, you know the protein structure and then you know the enzyme binding site. So if you know the protein structure, you can always, the lock and key mechanism, you can always decipher the enzyme binding site. And then there are drug screening libraries which gives you the drugs on the basis of the size, hundreds and thousands. And then there's the screening. So these things which I'm telling you, they need to be done in the simulation studies initially, where you get an estimation on the closest resemblance of a drug to your protein, and then you see the binding energies, how it is going to go and bind in that particular enzymatic site, right? So once it is done, then you can play with certain mutations in the amino acids. So then you can, after the mutations and everything, you can structurally reduce it, and after you think you have gone the perfect binding energies and it is fitting in well, you can then translate to your biochemical assays and see whether it is affecting the downstream process or not. I think I'm able to explain it, but let me know if you have any confusion right now. I'm doing bachelor's. Could you suggest any books or resources that help improve overall understanding in bachelor's in biotechnology? Of course, your course will be segregated into biochemistry and immunology and enzymology and everything. So every subject has its core books. Biochemistry is Leninger, and then there's Trier, and they have the specific editions. I used to study Leninger and Trier, and then there is immunology by QB. So I still rely on those books, and now majorly on the research papers. So yes. Okay, so if you just have the protein sequence, like, so say you just have the amino acid sequence of the protein. So there is a software called AlphaFold. If you'll put the amino acid sequence in the AlphaFold, it will give you a tertiary structure of your protein, right? So from there you have the tertiary structure of the protein, and then you move ahead. Am I able to answer? Okay, what are the challenges and the limitations of the cryo-electron microscopy? So the challenges and the limitations is, the challenges is a lot has to be done before you go to the cryo-EM lab. The purification of your sample needs to be ultra pure, and it needs to be very homogeneous. So that is a task. I mean, in my case, I spent around 8 months on the desk before I moved to the lab. But then again, there are a lot of troubleshoots that happen in the lab itself. But the major thing that you have to work is to have a homogeneous sample of a high concentration. That is the major problem that the cryo-EM has as a challenge. And In terms of limitations, like I said, there are chances that your proteins can be hampered if they are not stored or handled properly. There are high chances if the liquid nitrogen, if the temperature throughout your the cryo process is not maintained, you're not able to get what you require in any sense. So maintaining all those conditions and sample purity, and this is a little bit challenging. Although since if I talk about the computing and the software wise, a lot has been done, which and a lot has been pipeline also now. So I think that can be addressed, but the major challenge lies at the bench and the the environment in which you do your sample prep. Okay. I hope you find the presentation interactive and interesting. I tried to put in the videos so that you can actually relate to, and I believe in seeing is believing. Okay. If anybody want to come and talk to me, they can come to the Kusuma School of Biological Sciences. I'm on second floor in Professor Manideepa Banerjee's lab. So you can come and contact me and ask about me from anybody in her lab. A lot of people work on this protein structure modeling and drug designing and discovery in our lab. So they'll be happy to interact with you and give insights to you in this thing.
Thank you so much, Dr. Pankhuri, for such an insightful and enriching session. It was really great, and the eagerness to contribute of yours towards this session is really great. And even your lab, I got a chance to work in the transmission electron microscope on
Really?
some alpha synuclein protein fibrils as a
Wow.
part of collaboration between Dr. Rathore and Professor Banerjee. So
Yes.
I got to, first-handly experience that how cool is that. Like the cooling system itself made me, really like it was like a dream come
Fascinating.
true where we were seeing the vapors of that
Yes.
condensation through that,
It
liquid
looks like a
nitrogen.
magic world when the LN is involved.
Yeah, so it was really like surreal for me to experience that. And thank you so much for giving such an insightful and enriching session. Like this
Thank
kind of
you.
knowledge is gatekept from, people out there. So
Yeah.
thank you so much. Your explanation of cryo-EM and the ongoing research in viral disassembly has given us a deeper appreciation and how structural biology shapes modern scientific breakthroughs. And uh, your presentation was really interactive. The way you put videos and put a lot of efforts into it, it is really commendable, and
Thanks.
we are really grateful. Yeah,
I'm very also happy to present at this platform because I always believe what you can change in the first year of your graduation or the second year of your graduation is really impactful.
yeah, indeed, and
Okay,
it is because of yeah, there
somebody
is a
asked me, can I get a summer internship? See, you need to come and ask Professor Manideepa Banerjee if she thinks your research aligns with her lab environment. She would definitely be happy to have you. Yes.
Yeah, so participants can reach out to ma'am through her LinkedIn. She's really active and open to collaboration on LinkedIn, and
Yes.
she accepted our request from there as well. So thank
Right.
you, ma'am, and thank you all the participants who joined us today and made this session really interactive as well. And
Exactly.
we look forward to more of such sessions in the Beyond Research Student Journal Club, which is a collaboration that is brought together by the ACS chapter of IIT Delhi and Biopractify. in a effort in a effort to improve and enhance the access to network and nurture the provide the students from outside IITs and other institutes with the nurturing environment and network that they lack due to the limitations of their current institutes. So we approach ma'am, approach us, and write to ma'am, and reach out to us, and we would be really happy to you know
Sure.
give more insights into this. So
More
thank you
insights and
everyone.
guidance. Yes.
Yeah.
Yeah.
So do not hesitate.
Yes.
And we would really appreciate your feedback form. And once you fill that, we will also release your e-certificates after that.
Sure. Yes.
So thank you, ma'am. And we can conclude the session. And it was really insightful.
Yes, thank you so much for giving me this platform to share what I'm learning, and I'll be happy to address anybody's any questions or anything. And you can reach out to me, like I said, on LinkedIn or email or the lab. I am here most of the days, so we can contact and have a word, right? So thank you.
Thank you, ma'am. And we look forward to more of such engaging sessions with you in future.
Right, right, right. Anything impactful that comes from my lab, I'll be happy to discuss.
Thank you, ma'am.
Sure. Okay.
Thank you,
Bye.
everyone.
Good evening.
Bye.
Bye.